and Peace and Wellness, Vaccines, and the Call to Care. I'm Reverend Dr. Hammett Evans, and I serve as the Director of Pastoral Care for Methodist Family Health and as the Chair of the Arkansas Conference Board of Global Ministries. We're pleased to sponsor this webinar along with the Arkansas Conference Board of Church and Society, which is chaired by Reverend Betsy Singleton Snyder. The Board of Church and Society advocates for health policies and social justice. The Board of Global Ministries works to strengthen health networks worldwide. As United Methodists, we believe that health care is a basic human right, as we affirm in our social principles. When we protect ourselves and our communities through preventive measures like vaccines, we're living out our United Methodist calling to love our neighbors. This commitment to community health reflects both sound medical practice and faithful Christian witness. Global Ministries serves as the lead agency for the United Methodist Church's Abundant Health Initiative. Since 2017, this initiative has reached over 1 million children with life-saving health interventions across 50 countries. Through this work, we've invested over $26 million. We focus on preventing diseases, supporting vulnerable populations, and promoting comprehensive health education, including vaccination programs. Dr. Laurie Barber will be moderating for us today. She serves as our Arkansas Conference Abundant Health Coordinator for the Board of Global Ministries, and she brings over 30 years of experience as an ophthalmologist. She served at UAMS and also in private practice in Central Arkansas. As our Abundant Health Coordinator, Dr. Barber connects our annual conference to Global Ministries' worldwide health initiatives. She helps bridge the gap between global health advocacy and local community wellness here in Arkansas. Dr. Barber will be introducing our speakers and facilitating our question and answer session. So, Laurie, I'll turn it over to you.
Thank you very much, Hammett. I am very thrilled to be the advocate now for healthcare initiatives. And I was asked by Betsy Singleton to set up a process that we could talk to people about their questions about vaccinations. We have heard so many people that are confused about where to go and who to listen to about vaccinations and about the illnesses that they protect us from. And I picked two specialists that could be very helpful in our quest for the right answers. First, I would like to introduce our virologist named Rebecca Nelson. She's actually a member of United Methodist Church in Little Rock. She received her master's degree in virology and immunology at University of Arkansas for Medical Sciences, and was then hired to establish a clinical virology lab, or the study of viruses, and immunological testing, which means our body's responds to things such as viruses, and helped establish immunology techniques that were used to support the ability of patients to maintain their organ transplants, critical things. After 20 years of managing both virology and immunology at UAMS, she retired from that, but didn't really retire because she taught microbiology then at the University of Arkansas Pulaski Tech. Rebecca, you're on.
I should have cleared my throat first. The first slide, is that up, please? Yes. Well, that's my title slide. Prevention is indeed worth a pound of cure. Next slide. Next slide. Thank you. This is a little overview of what I'll be talking about. I am going to talk about some historical milestones in vaccine discovery. Then I'll go from that to trying to explain the science behind vaccines. I'm not going to go into any depth. It's a pretty heavy subject. I will talk about the impact of vaccines on public health and the challenges that we have in the future. Next slide. So we'll begin with smallpox. Until the 1700s, well, actually a little later than that, but we had absolutely mankind had absolutely no idea what caused diseases, infectious diseases. required the development of microscopes that would allow us to see them. The discovery of viruses came along much later in the 20th century, but in 1796, Edward Jenner, who was a country physician in Great Britain, observed that milkmaids, the young women who took care of the cattle and collected the milk, observed, and they also explained to him that they did not have smallpox. Smallpox was rampant everywhere for centuries. And they explained to him that they did not have it because they had contracted cowpox in their care of the cattle. So he decided to explore this and see what he could do with it. He devised a research project, I guess, program. Unfortunately, it was with a young boy, but he exposed him to cowpox. And then following that, exposed him to smallpox sometime later after he had cleared the symptoms of cowpox. He did not develop smallpox, fortunately. His next group of subjects were prisoners and they were there for a life sentence or for hanging. And if they would try this procedure, they could be released from prison. And so they had a number of volunteers. They were able to study a fairly large number for the time. And they were first administered cowpox and then later exposed to smallpox. None of them ever developed smallpox. So that was the beginning of it. From there, he published his findings. Even though he was just a country physician, he had a good idea about how to propose experimental design for this and data collection. And his work was accepted by the Royal Academy. So he went on from there. This was the beginning of studying immunology as well as looking at ways to modulate smallpox infections. It was a true scourge at that time. Can I have the next slide, please? Thank you. How the vaccines work in the immune system. I should probably tell you that I did not mean to put this slide in here. I had another slide in mind, but we're going to make this one work. It's just designed a little differently than some. Immune system activation. Wait a minute, make sure that I have it. How vaccines work in the immune system. Okay. In unprotected infections, a patient can experience a full range of signs and symptoms. These signs and symptoms reflect the attempt of the body to get rid of the infection. The extent of the illness varies with each type of infection. In some instances, the patient will become immune after contracting a disease, but in others, the patient will always be susceptible to reinfection. For instance, if you have a strep infection, you can still continue to have strep infections. If you have had smallpox, you should not get smallpox again. Vaccines were a way of preparing the immune system to quickly identify and combat harmful microorganisms in an initial exposure. The adaptive immune response, what this should also say is that we have an innate immune response. In an innate immune response, if we are exposed to an organism, we have many resources in the body to try to flush it away. We have in urine and in blood, it will wash it away. The skin is a barrier. The cilia in the respiratory system is a barrier. So that works. But the adaptive immune system uses cells and antibodies to protect us. When one is exposed to microorganisms, there are mechanisms that take place to try to deter the infection. The adaptive immune response makes use of T and B cells and antibodies to stop the infection. Vaccines have prepared the body for an immediate response. So this speeds up the system. You don't have to actually contract it. You can have a vaccine that sort of tricks the body into displaying the signs of the infection. The immunological memory. Immunological memory is the ability of the adaptive immune system to mount a faster, stronger, and more effective response upon re-exposure to a previously encountered pathogens. This is what happens in nature, but vaccines are another means of accomplishing the same thing. So what happens with a vaccine is that you're preparing the body, you're exposing it to a weakened form of the, hopefully weakened form of the organism. And with that, your body begins to build defenses that will protect you in the future. If I could have the next slide, please. Thank you. Types of vaccines and how they work. There are 4 listed here, well, 5 actually. Live attenuated. This is one that isn't used much, honestly. It's a weakened culture that may contain live organism. So the potential is there for actually causing an infection in the patient. That's not something that we want to do. Most of the time we're using inactivated vaccines, and this is the safest type. The organism is killed and cannot cause infection. It may require booster shots from time to time. If you can withstand the live attenuated, you should never need probably a booster shot. But with inactivated vaccine, you probably will. Every year I had students who told me that the flu shot gave them flu, and I had to go back and explain this to them again. It does not. It's inactivated, and so therefore you can't catch it. Subunit and toxoid vaccines. Subunit vaccines just use a portion of an organism to create immunity. And toxoids, there are some organisms that create a toxin, and a portion of that can be used to produce immunity. Anyway, messenger RNA vaccines. These are somewhat new. They have been working on them for some time, but during the COVID outbreak, this was the first time that most people heard about mRNA vaccines, and it was a little disconcerting to a number of people. Messenger RNA is consistently present in our bodies. Messenger RNA is mRNA. So it isn't new to us. It isn't some strange subject. It is used to deliver genetic information that codes for a particular protein and that the body requires. It can also be used to prevent infections. mRNA vaccines produce a protein that's essential to the organism. Our bodies then produce antibodies that will attack the antigen. So it's a very small molecule. All it does is go to the DNA or the chromosome. It copies that information, it carries it to a ribosome where a protein is made. And that protein serves multiple, depending on the protein that's produced, can serve multiple functions. And one is a protective sort of function. Well, that's probably enough. I'm probably just confusing everyone, but that's how the mRNA vaccines work. They are safe, they are reliable. We do have to change them sometimes as the virus changes, but some people are afraid of it because it's new. It sounds new. It sounds frightening, I guess. But we have been working on it for a very long time. In about 10 years previous to this, there was the SARS outbreak in Canada. Some of you may remember. And the SARS is a variant of COVID. Canada had a very proactive means of stopping it in Canada, didn't let it get into the United States or any other country. They did not allow flights in and out, and it did. They held it in place and did not contaminate the rest of the world. But scientists have been working all this time using that SARS variant, and And when COVID came around, it was not so difficult to come up with something new. I just talked with someone recently that I knew who is not in a scientific area, and he said, well, he was never afraid to take vaccines, but he was a little afraid of the mRNA vaccine because it was so new and untested. And I said it wasn't new and it wasn't untested. So I got to give him my little speech here from this. Could I have the next slide, please? Vaccine development and testing procedures. Before a vaccine can be released, it's typically a very long process, and I don't expect that to go away anytime soon. First, there's preclinical research, usually performed with lab animals, mice, rats, so forth. And then that information is the product that you get from your preclinical research is refined and is used in an actual clinical trial. That can take a long time depending on how many subjects you need to have. And then later it has to go to regulatory approval. There are governmental agencies that go over it with a fine-tooth comb, and they may send it back to you and have you do more. But it is a lengthy process. And then afterwards, after the vaccine has been released to the general public, there is post-marketing monitoring. And that goes on for a very long time to make sure that there was nothing unexpected, something that didn't show up in the earlier research. Typically, this is around a 10-year process. It could be a little more, it could be a little less. But they're very thorough with vaccines. They are not released until they have been well examined. If I could have the next slide, please. Thank you. The role of immunizations worldwide. One of the issues is making sure that vaccines are available to other countries as well as here. We do have mechanisms for supplying vaccines in the United States, but sometimes outbreaks occur somewhere else, or you have a very vulnerable population there where it seems you have lots of cases of it because their bodies are not able to handle this organism. So vaccines are crucial for taking care of those patients in other countries and ultimately patients here. Public awareness. You have to make people aware that there is a vaccine and that it does work. I've answered many, many crazy questions about vaccines. There's lots of misinformation that goes around. And so every class period was an opportunity to reinforce that they are safe and they are well studied. And they continue to be studied because something could happen along the way to change that information, and people need to know it. The high coverage goals. We want most of the people that can take it to have a vaccine. And so there are programs for having that. There are any number of people in this country and in other countries who are vulnerable. They have underlying conditions. Some of them have autoimmune diseases. Their infants may not be prepared for a vaccine right away. Just the elderly sometimes have a very difficult time with it. So we need to be able to protect the people around us. And that's why having a larger vaccine program to make them available to the largest number of people that we can in the population is a way of protecting those individuals who cannot adequately protect themselves. Next slide, please. Next slide. Is there another one? Yes, there it is. The impact of vaccination on public health. We have been, in a few instances, and only a few, been able to actually eradicate diseases. Smallpox was eradicated in the late 1940s. The polio, for all essential purposes, has been, although it sort of pops up every now and then. After an earthquake in Haiti, all of a sudden there were polio cases. No one knew exactly. I've not followed that to see if they know now, but they did not know why and what that had to do with anything that happened with an earthquake. But it's something that we have to keep after if we want to we know that we can essentially eradicate something, so we have to keep watching it and testing it and so forth. Herd immunity, this is an important concept that's been around for a long time, but if you want to protect those people in our environment who have diseases, if they're undergoing chemotherapy, their immune system doesn't function as well as it should, they're very young, they're very old, any number of things. The way that we can protect them is by protecting everyone else so that the disease just isn't available to them. It's a numbers situation. And it does work if you can promote vaccines. Protecting vulnerable populations, well, once again, I just mentioned that in the other three. Vaccines play a critical role in addressing worldwide health challenges and preventing disease outbreaks. Yes, it does. And you never know where something's going to crop up. I remember the AIDS epidemic. It was a very big deal. It happened just about the time I started working in clinical medicine. And if we did not have governmental agencies keeping tabs on things, they just happened to notice there was an extraordinary number of patients with fungal infections, bizarre pneumonias, one thing and another, and they were all in the San Francisco area. So they went into those communities and began looking and testing and discovered what they then named HTLV3 and later named HIV. And if they had not kept track, had not performed surveillance in different places, this could have gone overlooked, and we might not be at the point we are today where we can pretty much manage it. So I believe that's the end of my talk.
Well, thank you, Ms. Nelson. That was very informative.
Yeah.
It's good to hear some of the background and the science about vaccines and viruses. And I appreciate you telling us about that. If there are any questions related to Ms. Rebecca Nelson's discussion, we might be able to get to that after Dr. Sharkey's. And I would like to now introduce Dr. Marty Sharkey, who was born and raised in Fayetteville, Arkansas, and graduated at the Fayetteville High School as valedictorian. She attended undergraduate at Rice University in Houston and engaged even then in research on the then newly emerging HIV epidemic. Dr. Sharkey then attended Vanderbilt University School of Medicine in Nashville and pursued her academic interest in both infectious diseases as well as vaccinology, or the science of vaccines. She was awarded the Merck Award for Excellence in Infectious Diseases based on her research into tuberculosis, the BCG vaccine, hepatitis B, and the hepatitis B vaccine. After receiving her medical degree, or MD, she completed her internship and residency in pediatrics at Johns Hopkins Hospital in Baltimore, where she continued to pursue research in vaccines, including participating in research into the respiratory syncytial virus vaccine, which is the RSV vaccine, which we've been hearing so much about for babies and adults. She returned to her hometown in Fayetteville, joined the Northwest Arkansas Pediatric Clinic for many years, and then opened her own pediatric clinic. She's remained involved in vaccine research and was actually the lead investigator for the first specific meningitis vaccine to come to market. In addition, she was the first pediatric director for UAMS Northwest Arkansas and helped to start the first medical school in Northwest Arkansas. During this time, she also attended Emory School of Medicine with all of her free time to become a master in public health. Her thesis at that time was on the media's impact on MMR vaccine hesitancy. During the very early stages of the COVID-19 pandemic, Dr. Sharkey was called on by the city of Fayetteville, and I saw her also on the statewide news programs, and she discussed local epidemiology of the pandemic, the public health messaging of the pandemic, as well as leading mass vaccination efforts. She serves on multiple COVID-19 task forces at both the local, regional, and state levels, and has subsequently been recruited to work as a consultant to the CDC in the Immunization Safety Office. Her work throughout these years have led to numerous publications and multiple awards, and we are so excited that our own homegirl is now able to talk to us more about vaccines and their importance. Dr. Sharkey?
Thank you for that kind introduction. I'm going to try not to repeat too much of what Rebecca said, and try to build off of it. So vaccines, as we know, have a long history of success protecting people, individuals, and communities against infectious diseases. Just in the past 30 years alone, vaccines have prevented over 18 million hospitalizations and over 1 million deaths worldwide. They've improved the quality of life for many, and serious diseases such as smallpox have been eliminated. And as the technology advances, researchers can continue to develop better and safer vaccines. So looking at the safety issue right now, which seems to be a hot topic in the media, I want to talk about what's in a vaccine. So in addition to the ingredient, the inactivated virus or live attenuated bacteria, so that component that's the active component that's supposed to boost our immunity to induce us to make antibodies to fight a particular disease, there's a few other things in there, and I think a lot of people get worried about what are those few other things. Everything that's in there serves a purpose. First of all, they need something in there to keep the vaccine safe and long lasting. You obviously don't want it to be sitting on a shelf and get moldy or destabilize. So we need something in there to prevent that. And then sometimes with just the active ingredient alone, we don't see a robust immune response. So sometimes there's something called adjuvants that are put in to help boost the body's response. So there's stabilizers that make it more effective and stabilize the shelf life. An example of that would be gelatin or sugar. The adjuvants to help boost the body's response, aluminum salts are often used for that. Sometimes there's some residual cell culture materials. For example, we all hear if you have an egg allergy not to take the flu vaccine, and that's because the virus that is inactivated for your flu vaccine is grown in egg cultures. And so sometimes there's some egg protein that is in there. So you don't want to accidentally expose somebody who's allergic to egg protein by injecting it straight into their arm. That's bad medicine. There's some antibiotics that are put in there to keep other bacteria away. And these are common antibiotics such as neomycin or streptomycin. Penicillin is not used. Too many people are allergic to it, so we avoid that. And then if there is a vaccine in a multi-dose vial, such as we used to do very commonly with flu vaccine, sometimes there is a preservative in there, and a lot of times it was thimerosal, which does contain small amounts of mercury. We've heard a lot about this in the news. Only now, if you're taking a multi-dose vial from a flu vaccine, is there a potential that you're getting thimerosal? And those are pretty much now going by the wayside due to the most recent recommendations from the CDC. For years, we have not been giving those to children. We've been avoiding any preservative containing vaccines in children. The one other type, and I just want to mention it for completeness sake, is that some tetanus TD vaccines also have a little mercury in them. However, most people now aren't just getting a plain old tetanus shot. They're getting that with the pertussis booster, which doesn't have any. So those are also going by the wayside. Rebecca went into how we go about developing a vaccine. There's first a proof of concept. We tried the vaccine in an animal model. Then we moved to a phase 1 study, which is a small group of people are given the vaccine basically to make sure that it's safe. They aren't really looking for efficacy. They're just making sure that we can give this to people and the side effects are safe. Then in a phase 2 trial is more proof of concept. We give this to a bigger group of people. It's safe, and does it provide any immunity? Is it doing what we think it's going to do? If it does, then they move to a phase 3 studies, which is where thousands and sometimes tens of thousands, especially in the case of the flu-mist vaccine, hundreds of thousands of doses were given in the phase 3 trial. And this is to make sure that the vaccine works. They monitor common side effects, start looking for rare side effects because you have large enough numbers to start seeing those, and document safety. All that information is then provided to the FDA. And then if it gets approved by the FDA, all vaccines undergo phase 4 studies, which is post-licensure studies, as it's being given to the general population. And this is a formal ongoing studies to evaluate the new vaccine safety and effectiveness over quite a bit that the government is doing to look at those during those stages. So first of all, there's a passive surveillance, which is the VAERS system, which you may have heard of, the Vaccine Adverse Event Reporting System. Anybody for any reason can go online, fill out a VAERS report and say, I got this vaccine and this happened to me. It can be, I got this vaccine and I got a sore arm. I got this vaccine and the next day I turned purple. I've seen it all on these reports. Or it could be, I got this vaccine and I got really sick, and we need to look at those. Then there's the vaccine safety data link. This is a active surveillance system performed at multiple big medical centers throughout the country, where they are actively following people after vaccination who are getting kind of free medical care as a part of being enrolled in these use of the vaccine safety data link. So we're following these people for weeks and months after the vaccine to see if there's any side effect. And then there's the clinical immunization safety assessment project. And this one is where, say you get a vaccine and you get something really weird, you get the vaccine and then a month later you develop some new autoimmune disease, say. CISA would be the one to really do a deep dive on that and say, okay, is this vaccine related, or did this happen by coincidence, or can we figure it out? So we're always on all vaccines continually monitoring the population for any potential side effects, any new signals, anything at all. So the ACIP I want to talk about for a minute, going back a little bit. So after the FDA approves a vaccine, after the phase 3 trials, all that information, if FDA approves it, it goes to the Advisory Committee on Immunization Practices. And their job is to decide how safe and effective the vaccine is when given at specific ages. Should we give this vaccine at birth or at 2 months? That was the big happy conversation this week that they tabled. how serious the vaccine preventable disease is. For example, when the chickenpox vaccine came out, everybody's like, you get it. Nobody gets that sick from it. Why are we vaccinating against it? You know, you get the disease, you may have some scars, you move on. That was the one vaccine that was actually licensed, not because the disease was so horrible, though the sickest kids I've seen have had varicella pneumonia. Um, it was because for those of us who grew up with chickenpox, remember, you have to isolate at home for 14 days. The caregiver has to be home with you. So there was an economic impact of varicella, that they felt that the economic burden of the disease was worth vaccinating. It's the only vaccine where they looked at the socioeconomic or economic portion of it versus just the disease burden. Interesting. I think it is, at least. So I want to talk then about some of the side effects that we see with a few vaccines that have been in the news lately. I'm going to start off with the hepatitis B vaccine. Hepatitis B vaccine we give at birth, 2 months, and 6 months. Everybody says it's a sexually transmitted disease. Why are we giving it at birth? Well, for a couple of reasons. One, it can be transmitted in the birth canal. So there can be maternal to fetal transmission of the vaccine or of the virus. So we definitely want to protect all the babies whose mothers are hepatitis B positive, and you can be hepatitis B positive and have no idea. So that's number one. Number two is that we have seen that there does seem to be some household transmission of hepatitis B, whether it's from minor nicks and cuts or just living in a household with somebody who's hepatitis B positive, be it the mother or somebody else, and then children are testing positive for it. No clear reason that they're getting it at young ages. So, you know, they aren't IV drug users, they aren't sexually active yet, but they were negative at birth and somehow got hepatitis B during childhood. So it's a very important vaccine. We have also tried to just target high risk individuals. So we say if we're a man who had sex with men, or if the way we said it at the time, or an IV drug user, you should get the vaccine. Well, guess what? Nobody came into the clinic saying, oh, that's me, give me the vaccine. Didn't happen. So it continued to spread. We've seen a remarkable decrease in hepatitis B in the US since we've had this license. And the crazy thing is that hepatitis B virus causes a chronic infection that can cause hepatitis cancer, hepatic cancer, cancer of the liver. And we've seen that drop off as well. So this is the one vaccine where we've really seen a dramatic, well, also the HPV vaccine, dramatic decrease in cancer. So the people, it can cause a fever when you give it, like most vaccines. And so people are worried about when we give it at birth and a neonate has a fever, is that more dangerous to the neonate? Should we be avoiding that? The science right now says no, but there's still some discussion going on about that. The other one that will be coming up, ACIP is going to be talking about at their next meeting, is the Gardasil vaccine. The one thing we have seen with it, this is a vaccine against human papillomavirus given to teenagers, young teenagers, and we see a lot of them faint after they get it. It's a common thing. So they're going to be looking at a little bit of that. I think there's going to be a lot of discussion about that coming up. And then the flu vaccine, as Rebecca mentioned, people say, oh, well, I got the flu from the flu vaccine, so I'm not going to get it again. What you got was your body trying to learn to mount an immune response, which is what we want. So your arm's going to be sore. You may run a little fever. You may be a little tired. That is your body learning to fight the virus. So yeah, do you feel a little bit like you have flu symptoms? Yes. Do you have the flu? No, kind of the same thing we have with COVID. And then last but not least, the MMRV vaccine. MMR and varicella are those live attenuated vaccines that Rebecca mentioned. With these, we do see more side effects. And when they are combined together, we see a higher fever. And as most of us know, when kids get fever, it's not uncommon that they have a seizure. And we were seeing that with the MMRV vaccine when it was given at 12 months. And so the ACIP recently recommended that it not be given in a combination, that the MMR be given separately and the varicella vaccine be given separately. And actually, that's what I'd done in practice for years, just because I didn't like getting phone calls in the middle of the night with people having high seizures and fever. Wasn't the only reason, but that was part of it. But I would do it at the 4-year-old visit, the pre-K visit. I would do the combo vaccine, because then you have a terrified 4-year-old in front of you, and the fewer needles you use, the better. And at that point, their immune systems were more mature, and they were less likely to mount a higher fever or have a seizure at that age. So I want to also talk a little bit about herd immunity. Why do we all need to get a vaccine? Like, what does it matter to you if I don't get a vaccine? Use measles as an example. Measles is the most infectious disease we're aware of. If one person gets measles, and let's say the whole community is not immune to it, nobody's had a vaccine, they are going to infect 12 to 18 other people, and that's called the R naught. How many people a sick person will inoculate, will cause infection in and pass it on. And the higher that number is, the more people you need to vaccinate in your herd to keep it protected. So measles is our canary in the coal mine. That's when we know we are not having enough people vaccinated to protect the entire herd. Why do we want to protect the entire herd? We want to protect those that can't be vaccinated, those undergoing chemotherapy, those who do not mount a good immune response to the vaccine. I'm actually one of those people. I am rubella non-immune. I've gotten that vaccine 4 times. I'm Hep B non-immune. I've gotten that vaccine 9 times. I'm just not a good immune. I don't mount good immune responses to vaccines. The very little, infants at birth are protected from their mom's immunity. By 6 months, those antibodies are gone, and they are no longer being protected by their mothers, unless they're being breastfed. So protecting the least of these, I mean, this is what it means. And for me, as a Christian, as a Methodist, as a citizen, this is what you do to protect others. We've been weaponizing in the world viruses and vaccines for forever. We threw contaminated blankets gave contaminated blankets to the American Indians to give them smallpox. They used to sling dead bodies that were contaminated with the plague over barriers in the Middle Ages. When the first smallpox vaccine came out, Russia in particular started in the turn of the century, not this most recent one, the turn of the last century, did propaganda trying to get people not to get the vaccine. Why? Why did we vaccinate people back in the turn of the century? We wanted soldiers. That's why the flu vaccine was developed after World War I and the Spanish flu epidemic. So it has been trying to tell another culture to not, or another country to not vaccinate. It's been around for a long time, and it's still going on. We unfortunately are still seeing this today. We're seeing a lot of other stuff going on today, unfortunately. But I just hope that for all of us, as we look critically, think about what's best for us and best for our community, best for our neighbors, that we continue to promote good science, promote good health, and keep each other safe. And that is all I have.
Okay, I'm trying to unmute and start my video. Thank you so much, Dr. Sharkey. That was excellent. I hope that people that have questions are getting it to those that are managing the whole webinar. As I wait to hear of any questions, I want to mention that all of us as members of the United Methodist Church, even though our social principles do not explicitly mention vaccinations, the whole stance of the United Methodist Church is firmly rooted in its support for public health, preventative care, and equitable access to health care. So that's based on Wesleyan tradition of do no harm and do good to all we can at all times, every way we can, and also on the biblical principles of loving one's neighbors, as Dr. Sharkey had mentioned. Do we have any questions from the group at all? We have a few minutes, so if not, I've got a couple of questions for Dr. Sharkey. I'm proud of the fact that there has been a steering committee for the Vaccine Integrity Project. And that was set up and funded by daughter-in-law of Sam Walton here from Arkansas. And that the Integrity Project was looked at and included Asa Hutchinson, who is our prior governor for the state of Arkansas. So proud to have Arkansas connections to a project that's trying to comb through greater than 1,400 studies and figure out what it is we need to do here in the United States. And Dr. Sharkey, do you think the decline in vaccination rates may be due to misinformation? And if so, what percentage of that decline do you credit misinformation for?
Yes,
Roughly.
and I have no way of knowing. There is when you're a mother, I mean, we've all, as mothers, been there, and you're handing your infant over for their immunizations and you're watching this and you know that your baby's going to cry, you know that they may be miserable for the next 24 hours. It's hard. And so there is a sense of omission and sense of commission. So if you give your child a vaccine and they have a side effect, you have one of these things that you've been misinformed and scared to death of, you're like, oh, if I cause that, then I'm responsible. But if you don't vaccinate your child and then they get something, it's not you didn't actively do it. So there is a lot of, with this maternal fear and misinformation that is nudging that fear higher and higher. I can understand how a mom would be like, I'm not doing that to my child. I'd rather risk the disease than do that, because then it's a passive thing versus an active thing. So I understand it. And it gets scarier every day as more and more misinformation is out there in the public, and it's coming from within and without. We know that our adversaries are promoting it, and we know that there are some groups within the US promoting it. So we just need to keep fighting the good fight as best we can, and putting good science out there with good data, and soldier on.
What would you recommend your possibly pregnant daughter do about the vaccinations that are now being questioned?
I would, as far as the MMR and varicella combo, I'm fine with those being split up. Like I said, that's what I did in my
For
practice.
the grandchild, yeah.
Yeah, for the grandchild. I would want them to get the COVID vaccine, during their younger years. The HPV vaccine, when it's appropriate, I feel perfectly positive about it, and the hepatitis B at birth is also safe. So do I think I would I don't know what's going to happen when my daughter finally gets pregnant. But I don't know 100% for sure that I would recommend an annual COVID vaccine if they're 8 years old and healthy. I'd have to think about that, but definitely during the younger years, I would.
How about your pregnant daughter herself getting the COVID shot? You know, that had been
Absolutely.
previously recommended.
Absolutely. We have seen these, unfortunately, a lot of very tragic stories of women getting COVID during pregnancy, and just so sad. You know, as we've learned as a country, vaccines they're not 100%. You know, it's not, I'm giving you this and I guarantee that your body is going to respond and mount a 100% effective immune response. But what we can say is your chance of being hospitalized or dying from this disease if you're vaccinated is much less. So I highly recommend it, because I wouldn't want to risk my daughter's life or my grandchild's life by not vaccinating.
I have a
What about-
question.
Okay,
If
good.
that's okay.
Yes.
I wanted to ask this is along the lines of the flu vaccine. You hear people talk about, I may have gotten it too soon, or I may have waited to get it too late, and that the flu season just continues to get wider. And so, Dr. Sharkey, what would be your recommendation for a middle-aged person with uh, some occurring health situations to get their flu shot? Would you recommend it as soon as possible in the fall, or would you recommend that we kind of get into it and get the dose that's going to be more likely to be seen?
So I recommend around the 1st of October, no later than November 1st. It's rare that we see significant flu levels before Thanksgiving. Some years we see it rise right after Thanksgiving, but when everybody travels at Thanksgiving, then potential spread starts to occur. So it takes 2 to 3 weeks from the time you get vaccinated until you have good antibodies. So that's October 1st and November 1st is my window. And then that also means the dogma is, and what studies have shown is, in general, for the entire population, not for any individual, is that it provides you about 6 months of immunity, and that should cover you through the entire season. Some seasons we don't see the flu until February, and then it can go into April and May, in which case, if you had gotten it really early in the season and we're having a late flu season, you might want to go get a booster. And there have been years that I have done that. When I had gotten it, in September, as soon as it arrived in my clinic, and all of a sudden I'm not seeing flu in high numbers till March. I'm like, I don't really want to get it. I'll do a second dose in the spring if I'm worried that my timing was off.
Thank you.
All right. If there are any other questions from the audience, let me know. But in the meantime, a couple things that have come up in press and from patients, even in my ophthalmology clinic, is I'm not going to take an mRNA vaccine because it's going to change my DNA. And I know that Rebecca did speak about the mRNA vaccines, but could you tell us how dangerous the mRNA is to our own DNA?
Oh, it's not. So the way the mRNA vaccine works is it's messenger RNA, which when it goes into the cell, it interacts with what's called a ribosome. And the ribosome uses that mRNA as directions. Think of it as a recipe to make a protein. And in this case, with the COVID vaccine, it's the protein that looks like the COVID spike, the little green virus that we saw for forever. And then the cell that the mRNA has been working in will actually have those little spikes on its cell wall. It doesn't have a cell wall, but and your body will start making antibodies to it. It'll be like, oh, that's foreign. We don't know that. We need to make an antibody against it. So it works outside of the nucleus, interacts just with the ribosome, and then after it detaches from the ribosome, they have to connect to knit this protein. That mRNA floats off into the cytoplasm, biodegrades. The cell is very good at excluding waste. It gobbles it up and recycles it, and it's gone. So it does not interact with your DNA. So there's no fear of that.
That's important for people to know. I also think there is some discussion about getting rid of mRNA vaccines and mRNA technology. Can you address some of the other uses that we've had for mRNA?
So exciting, and it's breaking my heart. Our big area is in cancer research, and we have seen very successful mRNA vaccines for specific cancers. So basically, we can program that mRNA to make any protein we want. So we can design it to produce a protein that looks like a protein that a cancer-forming cell does. And then we use the body to make its own chemotherapy. So those antibodies are now going to attack the cancer cells. And it's a very exciting area, and it breaks my heart that we're potentially losing funding for that area of research, let alone what we could do. We need to keep advancing that research. We don't know when our next pandemic is going to occur, what it's going to look like. We don't know when the next Ebola or SARS, let alone COVID, it is an exciting, crucial step to us protecting ourselves, and I'm very saddened.
Hopefully we can get some more research money towards that technology. Speaking of research, one more thing, and we're about ready to run out of time, but there's been a key phrase that's being used now that we're going to start using gold standard research. We're going to start using, I know I've done a lot of research in my time, and that you are like the queen of research right now.
Well, one thing that we hear a lot of is that a double-blind placebo-controlled study is the gold standard, and it is. Except for when it's unethical. And in this case, when we're talking about not vaccinating a child against a disease, so they're in the placebo arm and somebody else is in the vaccinated arm. Let's say we go back, and because what they're talking about is going back and redoing all of our vaccine research with gold standard research. Let's take the Hib vaccine, for example, the HIV vaccine. This causes Haemophilus influenza B, causes a very serious meningitis in children. We've seen it be effective. It is unethical for us as pediatricians to first do no harm, to not vaccinate a child with a vaccine that we know can prevent a fatal illness. So we don't do those studies. It's kind of like what Rebecca was talking about, and kind of go full circle here to conclude. She was describing those smallpox studies that Dr. Jenner did. They would not be allowed today. You could not vaccinate a young boy with the cowpox and then expose them to smallpox, which we know could have killed him, and he had no proof of concept. I mean, there are ethical ways of doing research that sometimes the gold standard and the ethics clash, and we have to then do the best, most honest, safest work we can.
Very good. That's perfect. And everything's phased in So you start with non-humans and then go to humans ethically.
Right.
Thank you so much. Excellent.
Thank
Does
you.
anybody want to walk us out? Hammett, are you still around? Or Betsy?
I'm still around. Thank you all for coming today. We appreciate all of our panelists, and Betsy's there waving too. So thank you so much, and we appreciate your participation. And this will be available for sharing online. The webinar was recorded, and so if you have a friend that wasn't able to attend today, we encourage you to share it with them as well. Thank you all, and God bless.
Thank you.